Thanks, Tanya, and thanks for all of you for coming to hear my talk today. I will say that, my lab is a young lab. We've been at Santa Barbara for the past 4 years, but really with the help of the Department of Energy and the JGI and MSIL, as I'll describe, we've really been able to crack open a really difficult set of fungi that have biotech relevance, particularly for biomass degradation, and that's what I'm going to tell you about today. Okay, so Tim highlighted this very nicely, what motivated us to look at these unusual early branching anaerobic gut fungi, and that's really looking for new methods to try and get at this problem of using non-food sources of carbohydrates in order to accelerate this extraction of the sugars, and then converting those sugars through microbial fermentation into value-added products. And this could be a fuel, it could be a commodity chemical, it's probably all of these things, right, in an integrated process. And as everybody probably well knows, is that this is a really complicated substrate, right? This is something that not only has cellulose, it has hemicellulose, it has lignin, right? A lot of interesting chemistries already built into lignin, but there's also pectin, there's other biopolymers. All of these things are wrapped up all together, and we need to access them, separate them if possible, and then break these down or not, depending on whether we want to build up chemicals or actually use some of nature's chemistry in different ways. And so in industry, how this has been done is typically through expensive pretreatment and the addition of enzymes. And this has been helpful, but I would argue that there's a lot that we can learn from nature in terms of finding better enzymes. And we've already heard a little bit about genome mining, and I'm going to talk a little bit about that today. But this is really where my lab is focused, is trying to go from this step of really complex, non-food, lignin-rich biomass into sugar by eliminating pretreatment or greatly reducing it and trying to find better enzymes that are hidden in nature. And so to do this, we're inspired by nature. If you look around, you're going to see a lot of microbiomes that have learned through millions of years to turn over crude biomass, right? And not surprisingly, one of these locations is in the rumen of an herbivore like a cow, or in the hindgut of a horse, and large herbivores, right? And there, you're not going to find one particular microbe that's doing all the work. You're going to find a community of different microbes, and these anaerobes are really, really great at working together, dividing and conquering the difficult steps of biomass hydrolysis. And how we look at them as engineers, as many of you probably also do, is that each one of these microbes plays a role in the community. Some of those are doing the business of breaking things down. Others are helping those active microbes do their job by wicking away things that might be toxic or hazardous to them. Others are opportunists, and they're there to just kind of take things that might come off of the hydrolysis process. But the overall point is that each microbe has a job. It's there for a reason, and there's a lot of dynamics associated with these communities. And it's now really exciting to be doing research in this area because of the omics-enabled tools, not only genomics, but also proteomics, metabolomics. A lot of things are becoming more accessible to us, and integrating those techniques is really important. And so what we want to do is to really learn from these anaerobic gut systems how we can not only identify enzymes, but find natural systems that we could harness for bioprocessing, even getting towards synthetic consortia that are inspired by these systems. And so in doing that, we've really started by focusing on the anaerobic gut fungi that you see here. So these are colonizing wheat straw, and you can see they kind of look like little squid-looking things, and their rhizoids are penetrating into the biomass. I'll show some of their growth structure in just one slide. But what's really exciting about these guys is that they can grow into completely unpretreated biomass without any sort of help. They're going to do it slow, but they're going to do it. And so we want to understand how they're performing these functions. So just to continue on the discussion from yeast into fungi, right? So fungi are really interesting. There's a lot of people in this room that work on fungi, and I don't have to tell you why we love them, but we can learn a lot from their diversity, right? So we're focusing on the Neocalymastico mycota, which are very early branching. I've pointed them out here, or we'll affectionately call them the Neos. There's 6 fairly established genera or flavors of these fungi, although there's more and more seemingly every year as there's a greater community that's looking into these guys. And they grow in sort of a weird way, right? So they start off as a single zoospore, and they kind of swim around in solution or in the gut system until they find a piece of biomass. And then once they're happy and they found a good spot, they get rid of that flagellar network, and then they start to germinate into a rhizoid, more mature-looking fungus. That is where really the business of biomass breakdown is happening, as there's enzymes that are shot out from this root network, and the structure grows and grows until it becomes packed with zoospores that burst and liberate, and you have zoospore release again. And so this is kind of cool, and you can see why this is probably difficult to study. Another reason is that up until fairly recently, there's been a lack of genomes and available strains. If you want to work with these organisms, you generally have to go out into the world and get them, which I will tell you about. They have complex anaerobic growth. You can learn how to work with them. It's not that hard, but it's not something you can easily read about in a paper. They're difficult to cryopreserve. Actually, we and others have been trying to combat this issue. Currently not genetically tractable, no proteomic characterization, and there's a small but very hardy group of researchers that work on these fungi, and I'm hoping that there'll be more every year. But in other words, they're very complicated, and so a great place for an assistant professor to stake her whole career. Okay, so why else do we care? So the enzymes, we're after the enzymes, right? And aerobes spit cellulose-degrading enzymes out into the environment, and they usually do so in a very high capacity and a very concentrated manner, but anaerobes are greedy, largely because ATP is very costly in these systems. You've probably heard of cellulosome-like structures from anaerobic bacteria. I'm going to tell you a little bit about similar structures that we and others have identified and characterized from the anaerobic fungi. And the main point here is that rather than having freely diffusive enzymes, all of the steps that you need are held together on a protein scaffold through these modular domains called cohesin and dockerin. And we know that these are important in fungi because fungi also have fungal dockerin domains that I'll tell you a little bit more about. And there's megadalton cellulitic complexes that we and others have found from these anaerobic fungi. And this is an extensive network of 400 or so different dockerin tag genes, some of which are CAZymes and others are not, in the genomes of these anaerobic fungi. But the structure, the composition, the enzyme tethering mechanism of these fungal equivalents of cellulosomes is still largely unknown. And so what I want to tell you about today are ways in which my lab is trying to contribute to ways to accelerate the production of renewable fuels, sustainable chemicals from non-food biomass and from agricultural waste. And to do this, I hope I can convince you that we're really learning from nature to engineer enzymes, anaerobes, and even consortia with novel functions. And so to do this, I have the story broken up into 3 parts. So the first, I would argue, is the most fun, and we're before lunch, so you'll see why it's good that you hadn't eaten anything. We go out and we extract these things from nature, and this is fun. And then we use omics-enabled tools, so genomics and transcriptomics for one, to discover novel enzymes from these systems and how they're regulated. And then finally, what I'll tell you about are ways in which we've tried to kind of marry the sequence data together with proteomic characterization to get at this problem of fungal cellulosomes. Okay, so here we go. So where do we go to get these guys? If any of you know Santa Barbara, you know it's a beautiful place. You may or may not know that we have a wonderful zoo. And so this is my lab heading off into battle to go find some nice anaerobic gut fungi from herbivores at the zoo. To do that, we have a great collaboration with the folks that care for the herbivores at the zoo. Here they are here, getting educated on what's going on. Here they are with my collaborator, Mike Theodorou, who's actually scooping up some fresh samples. And then I love this picture. This is what happens when you can take a picture of your group staring into the dumpster worth of of elephant dung at the zoo and sampling. So this is Sean Gilmore, who's carefully doing that, who's also here at the meeting. Fortunately for Sean and others in my group, you don't need a whole bin to extract fungi, and you only need a really little bit. And so how we do it is we use very old school microbiology enrichment techniques. So we start with these fecal samples, and then we grind them up, we dilute them. In our case, we're after the fungi, and so we kind of coax them out of this habitat by challenging them with biomass, but inundating with antibiotics. And how we separate them is kind of the equivalent of an anaerobic petri dish. It's called a roll tube. So rather than, these are anaerobes, so rather than spreading them out on a dish, we actually fill this headspace of a closed vessel with CO2, and the outside is coated in nutrient-rich agar that has cellobiose in it, where each one of these guys is a kind of an isolate equivalent or a colony equivalent that's been started from a single fungal zoospore. And so we pick one of these off the side, and we figure out what we have. And then we can characterize the strains. And so this is just a snapshot of some of the biological diversity that we've been able to capture from the zoo animals in this way. These are all completely novel because there's just a small community of researchers that's working on them. Here you see the head structure of this Pyromyces finis that I'll talk some more about. The root structure is penetrating into the biomass. This was isolated from a horse named Huckleberry Finn, and so we named it after the horse, naturally. This one, this Neocalamastis californiae, which you can tell who that's named after, has a much more circular structure. The rhizoids are very difficult to see. And this Anaeromyces is a polycentric anaerobic fungus that has 2 centers of growth. And it affectionately, we call it the whale tail fungus, and so as such, we named it after the gray whale that migrates in and out of the Santa Barbara Channel, Anaeromyces robustus. It's also coincidentally the most robust fungus that we have. Okay, so this is neat. We can get these fungi out of the environment. What do they do? And so we actually start in the lab by challenging them on diverse sources of biomass. So these will be everything as simple as sugars and simple starches on up to cellulose, avicil, reed canary grass, switchgrass. Long story short, for each of these isolates, they all have a preference in what they like to eat. This particular one from the horse, this is the horse, and this is unfortunately not me, but this is the horse. And you can see that they grow very well, high pressure accumulation on reed canary grass, and then equally well on glucose. This one struggles a bit more on xylose compared to some of the others, and I'll get back to that in a bit, but we could basically use this to characterize the behavior of the isolates that we're getting from nature. And so what I would say, just to echo some of the comments yesterday, that I realize I'm continuing some of the themes of talking about poop and then also publicity, but as a young assistant professor, you have to watch who you talk to as well. So we talked to the LA Times a few years ago, who were really interested in what we were doing. I will say that for sure they do quote you, and for those in the front row, you can read this. Those of you in the back, sit closer to the front next time. Okay. So this is kind of fun, but I think it's good because the country is interested, right, in this idea of taking waste material into value-added things, fuels, chemicals, whatnot. And not only our work, but everybody's work in this room, captures the attention of the public, and I think that's also very, very important for our mission. Okay, so looking at what we can do next. So we've isolated these anaerobic gut fungi from nature, and they do some pretty remarkable things, especially hydrolyzing reed canary grass, switchgrass, corn stover, et cetera. So what can we learn about how they're doing this, right? And so I mentioned that there were no existing genomes when we started looking at these anaerobic fungi, and even if there were, we have novel isolates that nobody's ever seen before, so we have to do a lot of building from the ground up, and we've done that largely with the help of the JGI. And so to begin with, just because it was easier, we actually started with transcriptome data. There's lots of reasons why the genomes are difficult. I'm going to come back to that. But essentially, what's nice about the transcriptome is that it captures the dynamics of protein expression and especially enzyme production. And so if we're looking at just kind of the complete coding sequences of the expressed genes, you might want to start with the transcriptome. And since we're looking for enzymes, we thought that we would explore a hypothesis that should be pretty straightforward, and that pretty much boils down to microbes are greedy, and fungi should be greedy too. What I mean by that is that if you have a culture and you force them to make biomass-degrading enzymes by challenging them with lignocellulose, they should make them, or else they're going to die. And so if you sample the transcriptome at that time during enrichment, you should see with next generation sequencing, you should see a huge wealth of the biomass-degrading enzymes. Conversely, if you start with that same isolate and you instead give it the equivalent of candy, like glucose, there's absolutely no reason for an anaerobe in particular to divert valuable cellular energy into making enzymes. And so instead, if you sample the transcriptome at that time, you should see a de-enrichment in the biomass-degrading enzymes. Of course, it goes without saying that such an approach is actually not that simple. It requires a lot of de novo assembly, annotation, a lot of bioinformatics that went into this. But needless to say, let's start. And so in working primarily with Igor Grigoriev's group, the JGI, we were able to assemble de novo transcriptomes without reference genomes. And this just gives you an idea of how we did this for this Puromyces isolate. We get about 27,000 transcripts. We built a pipeline to extract the ORFs and figure out through homology modeling if we had something that looks, at least initially like a CAZyme or like anything else that we can annotate. In doing that, we can only annotate about 1/3 of the transcriptome, which is not entirely surprising given the lack of representative genomes in this area. If we take a step back, though, and we count the CAZymes and look at the fungal tree of life, we saw something that was quite remarkable. And this is work that was just published in Science I'm going to tell you a little bit more about. And that's, essentially these genomes and the transcriptomes in particular in this case are packed with biomass-degrading genes. So much more so than some of the other fungi that are more evolved like Trichoderma. That's not to say that these are necessarily the best degraders, but they do at least have more machinery to handle more diverse inputs. And so this was pretty remarkable. In addition, they have actually, it looks like they've obtained a lot of these CAZymes from bacteria through horizontal gene transfer. What this shows, and again coming back to this Dockerin sequence, is that a lot of the genes are Dockerin tagged to participate in this fungal cellulosome complex. A lot of them are CAZymes, but a great majority of them seem to have come over from bacteria, rumen bacteria in particular. And I'm not going to spend a lot of time on this, but it will suffice to say that actually, we found in several cases that there's high homology of a bacterial-looking gene in the fungal genome and transcriptome, but rather than where there was a bacterial Dockerin before, now it looks like in some cases the fungus has completely gotten rid of the bacterial Dockerin and added a fungal Dockerin. And this is something that we're still trying to understand, but is very interesting. Okay, so I told you a little bit about the enzymes that we understand, that we can identify through homology modeling, but what about the enzymes that might be hiding that we have no idea what they look like on a structural level? And so getting back at this hypothesis that the fungi might tune their metabolism to handle different challenges, we looked at gene set enrichment of certain CAZyme families and glycosyl hydrolases just to see if that was in fact true. Perhaps not surprisingly, the fungi that we looked at tune their sort of fire their enzymes to match the target, the challenge that they see as we go from least complex, so glucose, really no challenge, to more challenging reed canary grass. You see more functional enrichment of certain cellulases, hemicellulases, it makes sense, right? And so we thought that maybe we could use this regulation pattern to pull out completely novel things. And so what some talented folks in my group did is captured, there's more details in this paper, but essentially we grew the fungi, the Puromyces isolate in this case, on reed canary grass to fire the enzymes. And then we added in glucose, and we expected that as the glucose was taken up, we should see a rapid remodeling of the transcriptome as the fungus then shuts down the biomass-degrading machinery. And in fact, that's exactly what we saw. So we saw a lot of things go up in that time that are in red here. These are largely associated with metabolism reprogramming, stress response, whatnot. But we saw a remarkable number of things in blue, which are repressed as the sugar is taken up, right? If we look using sort of our known enzymes as tracers, we find almost all of them are contained within this regulon. But what's even more exciting is that there's dozens of transcripts of completely unknown function that are tightly co-regulated. And so we're in the process right now of working with NREL and Argonne on high throughput screening and then also structural biology aspects to understand if maybe there's completely novel enzymatic domains that have been captured by just kind of deciphering these endogenous regulation patterns from nature. In addition, after we looked at the transcriptomes, PacBio came online and it was more accessible. And this is an example of the one published anaerobic fungal genome to date. And these are really tough to sequence. They're pretty large genomes. They're highly repetitive. They're very AT-rich. It's very, very difficult to crack. And so this particular genome is in about 23,000 pieces. With the help of Igor's group, we were able to really improve on this. And this is largely due to advances in PacBio sequencing and the expertise of the JGI. We have, for instance, this pyromyces isolate down to about 200 or so pieces, which is a huge advance for us in being able to understand what else might be hiding in these genomes. Okay, so for the last part of the talk, I told you about how we've isolated some of these novel fungi from nature that do pretty remarkable things, and how we've looked at their regulation patterns through sequencing to try and understand what they're doing, or at least the base set of equipment that these fungi have. And so how can we use that information to try and look at these complexes of enzymes? And so I mentioned that fungi produce enzyme complexes. They have fungal Dockerin domains, but there's a lot of gaps in knowledge that we don't quite understand. And so one talented postdoc in my lab, Charles Heitzema, who's actually here, did is he really wanted to get at this protein structure-function relationship by leveraging some of the sequencing. And so Charles figured out a way to basically crash these complexes out of solution by exploiting their ability to bind to cellulose. And he separated them through size exclusion, chromatography. They're megadalton structures. These are huge. He can also kind of break up the pieces parts, run them on a gel, and look at different signatures. So this is what the banding pattern might look like of some of the enzymes that come off on reed canary grass. We can punch these out, do mass spec, and they all align with our transcriptome data and our genome data. Similar pattern on filter paper, but the complexes more or less go away when the fungus sees glucose, which is not surprising given the data that I just showed. But the question remains, what happens, how are these things assembled, right? I've kind of drawn this schematic, but we don't really know that that's true. We don't know what the Dockerins are binding to, and this has been a great debate in the literature for the past 20 years or so. And so what we wanted to do is to take a step back and leverage some of our sequencing data to get at this question. So before I get into what we found, I want to just take a second and highlight what is known at least about the differences between the fungal cellulosome complexes and bacterial ones. So one is that the fungal Dockerin domains are not species specific. I can isolate a fungus from the Santa Barbara Zoo, and somebody else can isolate a fungus on the other side of the world. You will see Dockerin domains that look the same. They might be completely different, but they will look the same. Or they might be different fungi, but they will look the same when it comes down to their Dockerins. And these boil down to about 40 residues that constitute what's called a single Dockerin. But the other thing that's interesting about the fungi is this single unit is often repeated on the N-terminus or on the C-terminus of different CAZymes and other genes, such that you might have 2 next to each other, which would be called a double Dockerin. You might have 3, which would be called a triple Dockerin, on up to maybe even 8 repeats. And we don't understand what the significance of that is. There's also not been a non-catalytic protein scaffold that binds and links everything together. That's been pretty elusive. And what we found, at least, is that all of the components that we've identified seem to have Dockerin domains. And so we thought that was consistent with what else has been published in the literature. I am also pointing out that there's been some nice mutational studies that have been reported in the literature from Harry Gilbert's group, where you can actually mutate some of these key conserved residues, and we know that eliminates Dockerin binding. What it's binding to, no one knew, but this is something that was helpful for us. So we have all this sequencing data. What can we learn from it? So we can go through and look at all the CAZymes in particular that have Dockerin domains, looking at what the repeat nature is and where it sits, if it's ahead of the CAZyme domain, or if it's behind, right, N or C term. And long story short, in leveraging these 3 genomic and transcriptomic data sets, we found that the predominant form on of the dockerin on the CAZymes is a double dockerin that's on the C terminus. And this was something that was helpful in trying to get at the interaction. So I mentioned that the anaerobic fungi are not genomically tractable yet. We and others are working on this, but what you can do is you can express some of these domains recombinantly in yeast, right? And so what Charles did is he took this predominant form of, or dominant form of the dockerin domain, this double dockerin, expressed it in yeast. He set up a simple affinity chromatography assay to try and capture things that might be sticking to that dockerin. And then we wanted to look for what's stuck, right? So this is what a total cellulosome protein might look like. And this is what was sticking. So we found things that looked like a GH48 and a GH6, which are coincidentally the most abundant enzymes or proteins in the cellulosomes, given our data and others. And so we thought maybe the dockerins are just binding to each other. And that's not to say that that's not happening, but this is where I was really happy that we had one of the first ficus programs that leveraged expertise at the JGI and at EMSL, because we at the same time had some proteomic support from EMSL that came back and said, you guys have this really huge protein that's in these cellulosome fractions. Do you have any idea what this is? And we started looking at it, like, whoa, this might actually be a scaffold. So this is a huge protein, 700 or so kilodaltons. We never would have seen this on an SDS page, no way, right? And it's got a secretion signal, it's got cell wall binding motifs, it's got a membrane anchor. It also has repeat regions, which I'm going to show you in a second. In addition, there's some of these repeat motifs that are found in the GH48 and in the GH6, so maybe there is some precedent for dockerin binding to other dockerin-containing things. This is also downregulated on glucose, so it's catabolite repressed, and has absolutely no homology to any other sequence in the NCBI database, only in the anaerobic fungi. And so as Charles was looking at some of this sequencing data, he found this emerging motif, and we are not going to say that this is a minimal cohesin, but it does look awful suspect, and it repeats several times over in these scaffold-looking fragments, and so we were happy that we started to decipher some of this. What's more, we found sort of related proteins that might be scaffolds in all three of these genomes and in the transcriptomes. And when we started mining the genome some more with Igor's team, we found shorter variants that have a lot of these repeat regions. And we wondered if there was more to that. So Alan Kuo also looked more deeply at the genomic data sets and found that there's actually some relationship and clustering of different fragments that sort of link these three genomic data sets together. And so we're really, really excited about looking at this further. But genomes are jam-packed with these putative scaffold-looking things. And so you're probably wondering, I'm calling these things scaffolds because we found them in the proteomic data, and do they really do what we think that they do? And the last piece of data that I'll show is trying to reconstitute this binding interaction with ELISA and then ultimately with SPR. And so what Charles figured out how to do is basically, you can't express this whole scaffold fragment because it's so large. And we don't have the genomic tools for the gut fungi, but you can express truncations or kind of scaffold fragments. And so in doing that, he basically whipped up an ELISA that has a scaffold fragment binding to a recombinant dockerin. And if there's binding, we should see signal. Essentially, in doing this with a range of different dockerins, he sees saturation type behavior, which we did later SPR experiments and show that we have about 0.7 or so micromolar affinity, which is not super high, certainly doesn't match the bacterial system, but this is also a recombinant system, so whether or not we're capturing everything is yet to be seen. The double dockerin also has a saturation type behavior. Interestingly, when we mutate some of these key residues that are known to modulate dockerin binding, we also see a drastic decrease in the binding to scaffold fragments. So we think that we have a pretty good handle on this interaction, and we're trying to explore explore this more using high resolution structure like NMR or crystallography. And so just to sum up, I hope what I've shown you is that there's a lot of promise in looking at these unusual early branching fungi. In particular, we've isolated a panel of cellulosome producing fungi from nature. We've found thousands of novel genes with the help of the JGI and through de novo transcriptome assembly to begin with. Just by looking at transcriptome remodeling, we think we found what may be not only novel genes or novel CAZymes that kind of look like CAZymes we know, but maybe even completely different ones that have been hiding in nature. And we think that we might have a parts list for assembling fungal cellulosomes, which has really been an elusive part of working on these fascinating organisms. It's not to say we've captured everything about them, but I think we're starting to learn more and really get at what's going on. And so, as I mentioned, we're working on all levels of this, looking at molecular interactions, synergy between bacteria and fungi, putting consortia together, and whatnot. And so with that, I will thank the people who did the work, many of which are here at the meeting. You probably have already met with them or seen their posters. This is all was initially funded through an early career award from BER and the DOE, and is also aided by the Institute for Collaborative Biotechnologies, which is a U.S. Army funded center. And with that, I will thank you for your attention and take any questions.